Only one day after the virus identification, diagnostic tests were available due to the advancement in molecular biology. The gold standard technique, called RT-PCR, was invented by biochemists in the 80s. An RT-PCR reaction has 70% accuracy, which means it can lead to false negatives. It has to be done in a lab, and it takes about 2.5 hours to amplify the genetic material of a virus, allowing its detection. It is very important to have widespread and faster tests to identify asymptomatic people, as they seem to be the Achilles heel in this pandemic. Only then, tracking the spread of the virus and calculating the true fatality rate become possible. To be able to assess the infection in millions of people, robotics is helping in automating experimental steps. Scientists from different disciplines have developed faster tests based on colorimetry, microfluidics, immunity markers, and CRISPR-Cas genetic editing techniques. These new tests, called Sherlock, were recently approved by the FDA in May 2020, and it gives the result in about an hour. Home tests similar to the pregnancy test would be available in the near future. When we encounter the SARS-CoV-2 virus, our immune system develops antibodies to neutralize it within 20 days. While the PCR is positive only when the virus is still present in fair amount in the body, the antibodies can last approximately from few months to several years. For some viruses, it can last for life. Immunologists are optimizing serology tests to precisely detect these antibodies in blood samples. Now, 4 months after the virus was identified, a COVID-19 antibody test has been granted emergency use authorization by the FDA. This antibody test, seemed to have high sensitivity and over 99% specificity for SARS-CoV-2 infection. These tests are crucial for 2 reasons. First, to understand our immune response to the virus and how long can one person be immunized. And second, to study the spread of the virus in the population, and based on that, advise on strategies and timeline for deconfinement. Decide whether we have got or not herd immunity, or should we go for shield immunity. The transmission rate or reproduction number, R0, is the average number of people that one person would infect. With the social measures, the rate of transmission, R0, can decrease from 2.5, which means one person can infect 2.5 people, to 0.6. One person can infect less than one other person, just by staying at home. Numbers are extremely important in tracking and understanding what's happening. This allows making estimates based on mathematical models and planning for best strategies like flattening the curve. Epidemiologists, they create models to predict the pandemic evolution, and then these models can be matched with the ICU beds capacity, for instance, to help decision makers decide when to lift a lockdown. Biologists investigated the COVID-19 virus attachment to human cells and found that it attached more efficiently than SARS does. Scientists suspect that the spike protein is activated by an enzyme very early on upon the viral particles formation, and this allows it to be more efficient. Coronaviruses have the largest genomes of any known RNA viruses. Their RNA is composed of approximately 30,000 bases compared to 14,000 bases for influenza viruses. On the other hand, coronaviruses have a proofreading mechanism to reduce mutation rate and stabilize the genome. They mutate at a lower rate as compared to influenza. By aligning the genomic sequences available in databases and other websites, geneticists track the emergence of new mutations over time and understand the origin and spread of the pandemic around the world. For example, early genomes differed only by a few mutations, suggesting a very recent emergence and an outbreak from one source rather than repeated zoonosis. Tracking these new mutations allows scientists to adjust the development of a vaccine that would be active against different strains with different mutations. It is important to note that many mutations have been seen to date with the thousands of genomes of SARS-CoV-2 that are available, but none of these mutations have yet been proven to have an effect on the infectivity or the severity of the disease.